All right. So when you're looking at cell communication, right, it's really, really important that you always go back to the three components of a signal transduction pathway, right? Component number one is always going to be reception. Component number two is always going to be transduction. Component number three is always going to be response. All right, so those are the three most important things to know about a signal transduction pathway. All right, so when we're looking at a signal transduction pathway, it's always going to begin with step number one, or reception, right? And reception is going to take place when a ligand binds to a receptor. Now, we talked about three different types of ligands, right? Ligand number one is going to be a neurotransmitter. Neurotransmitters are used in what body system? The nervous system. All right, so that's our first type of ligand. Right, our second type of ligand is going to be a hormone, and hormones are used in what body system? Endocrine system. Right, the nervous system and the endocrine system, we learned yesterday, are the two major body systems of communication. However, we learned that they play different roles in communication. The nervous system is responsible more for coordinating activities in the body. Coordinating activities like muscle contraction. The endocrine system is responsible more for regulating or controlling internal conditions. Right? Or we'd also say it's more responsible for regulating what process? Homeostasis. Right? So homeostasis is maintained through the release of hormones. Our third ligand is going to be the growth factor. And growth factors are released through paracrine signaling. All right. Now, if we just look at these chemical messengers, neurotransmitters and growth factors are both examples of local messages, right? They are local chemical messengers, meaning they are being released by one cell and they are traveling to another cell that is in very close proximity. The key with a local messenger is that it is not entering your what? Your bloodstream, which is exactly what a hormone does. Right? A hormone is a long-distance messenger. Right? It's a long-distance message. And what makes it long-distance is that hormones enter your bloodstream. They travel throughout your entire body, and they bind to any cells that have a what for it? A receptor. Good. Now, if I was to ask you any type of question that has anything to do with cancer, what type of ligand do you think that's going to involve? A growth factor, right? So anything that is dealing with cancer is going to include growth factors. Right? Because growth factors tell cells to do what? to divide, right? Growth factors tell cells to divide. And cancer is uncontrolled cell division. So growth factors or abnormal growth factors are in some way implicated in cancer, right? Always keep that in mind. So during step number 1, reception, right? The chemical messenger is going to bind to a receptor. All right, and this is what a receptor might look like. Remember that a receptor is a protein, and every protein has a unique shape. And its unique shape is what makes it extremely specific for the ligand that binds to it. Right? So it's really important to understand and to know that receptors are extremely specific. You might even say that they work through a lock and key mechanism, right? Meaning that the receptor is the lock and the ligand is the what? Is the key. And they fit together extremely specifically. Right? So each receptor is extremely specific for the ligand that's going to bind to it. Now, let's say, hypothetically, that we want to prevent a response from occurring. Let's say that you have a cancer cell, and that cancer cell is trying to communicate with another cancer cell. And we want to stop that growth factor from getting to the other cancer cell. How might we do that? How can we stop the growth factor from causing a response in its target cell? What can we do? Shay, what do you think? Go ahead. No, you wouldn't. No, it's a good idea. Think outside the box. How can we stop a target cell from producing the response? How can we stop the ligand from getting to the target cell? Laurel, what do you think? Yeah. What you're going to interfere with is step number 1. So if you are somehow able to block the receptor, you would be able to stop that chemical messenger from creating the response. Does that make sense to all of you? And the way that you can block the receptor is to use something called an inhibitor. So an inhibitor is something that would bind to the receptor, and it would prevent the ligand from being able to bind. So if you can use some type of chemical that can bind to and block a receptor, then that ligand would not be able to bind, and that cell would not be able to produce its response. Does that make sense to you guys? So that would be an effective strategy to prevent one cancer cell from being able to communicate with another cancer cell. Right? You block the receptor that the growth factor would bind to. Does that make sense to all of you? So when we looked at these receptors, we talked about 4 different types. Receptor type number 1 is the GPCR, the G protein coupled receptor. And it's called a G protein coupled receptor because attached to it is a protein called a G protein. A second type of receptor that we went over is an RTK receptor. a receptor tyrosine kinase. A third type of receptor that we went over is a ligand-gated ion channel. A fourth type of receptor that we went over is called an intracellular receptor. Now, if you look at the top three that I placed into boxes, these top three receptors are all located at the cell membrane. So the type of ligand that's going to bind to them needs to be a polar or a hydrophilic ligand. So polar ligands are going to bind to GPCRs, RTKs, ligand-gated ion channels, right? Because they are at the membrane. We gave an example of each of these. What type of ligand, what ligand did we give the example as binding to the GPCR? Epinephrine, good. So epinephrine binds to a G protein coupled receptor. What's an example of a ligand that binds to the RTK receptor? The growth factor. And finally, we said that neurotransmitters bind to ligand-gated ion channels. So these neurotransmitters, these growth factors, epinephrine, these are all polar ligands because they're binding to receptors that are at the cell membrane. Intracellular receptors are receptors inside of the cell. And we said that ligands that are going to bind to these need to be non-polar ligands, because non-polar ligands go through the membrane and bind to receptors inside of the cell. And an example of a non-polar ligand that we talked about is the steroid hormone. Steroid hormones bind to receptors inside of the cell. Right? Now, just jumping ahead, looking at each of these types, because I know they are important, epinephrine triggers what response in its target cell? Fight or flight. So epinephrine is responsible for causing fight or flight. Growth factors initiate what process? Cell division. Neurotransmitters function in the nervous system. And then steroid hormones, this is the toughest one. Steroid hormones regulate what process? Gene expression. If you remember, steroid hormones are responsible for turning genes on or turning genes off. So steroid hormones regulate gene expression. Are we doing okay so far? Good.
Do we have to know what epinephrine's from? Like neurotransmitters from the nervous system? We have to know
If you need to know the words that are coming out of my mouth.
Okay.
Alright, if they didn't come out of my mouth, you don't need to know it. Alright, if I didn't include it in one of the lessons, you don't need to know it. Alright? We're doing okay? Any questions so far? Alright. Step number 1, reception. The ligand binds to the receptor. When the ligand binds to the receptor, that is going to initiate step number 2, which is going to be transduction. Transduction is when the message is relayed throughout the cell. When the message is relayed, it's going to relay using a couple of different things. Number one, it's going to be relayed using protein kinases. And we learned that protein kinases get activated through phosphorylation. And we call this process a phosphorylation cascade. And we described it like the proteins are passing the phosphate from one protein kinase to another. So in order to do that, you need a source of phosphate. And that source could be either ATP or it could be GTP. It could be either one of those. Now, sometimes you have a molecule that is passing the message, which is not a protein. Anything that's going to relay the message that is not a protein is called a second messenger. It's called a second messenger. Any molecule that's going to relay the message, which is not a protein. And some second messengers are, number one, cyclic AMP, the most widely used second messenger in the body. You have calcium, Ca++, and you have IP3. These are all examples of second messengers used in the body. You do not need to memorize any specific pathways. You don't need to know GPCR to G protein to adenylyl cyclase to cyclic AMP to protein kinase A. You don't need to know that. You don't need to know GPCR to G protein to phospholipase C to IP3 to calcium to protein kinase. You don't need to know that. You need to know what protein kinases are. You need to know what second messengers are. This is then going to lead to a cell response. Step number 3. We talked about 3 general types of cell responses. Response number 1 is going to be a change in cell behavior. Right? A change in behavior. We talked about increasing heart rate. We talked about pupils dilating. See that? Right? These are all examples of changing the behavior of a cell. Number 2, we talked about activating or inhibiting enzymes. A really good example of this that we learned is the process of apoptosis. Right? In order to induce apoptosis, we activate certain hydrolytic enzymes like caspases, like proteases, like nucleases. These are the enzymes that are going to be responsible for breaking down and digesting the entire cell. We talked about 3 reasons that apoptosis would be triggered. Somebody give me one of them. Go ahead. DNA is damaged, so you have a mutation. A mutation is one cause of apoptosis. Sophia, give me another one. A protein is misshaped or misfolded. That can trigger apoptosis. And finally, third is if the cell receives a what? A death signal. Right? That can also cause apoptosis. And remember, apoptosis is programmed cell death. Right? Where the cell kills itself to save the organism. Right? Apoptosis is a perfect example of the activation of enzymes. Growing a shmoo would be a good example of changing cell behavior. And finally, our third response, the most important of those three, is going to be turning genes on or off. And remember, turning genes on or off is going to control our body's production of proteins. We know that proteins do everything. And we talked specifically about steroid hormones like testosterone and estrogen turning our genes on or off, controlling the production of proteins in the body. This is everything on cell behavior, right? The other thing that's going to be on the test, which I don't want to review because we went over it yesterday, is going to be the endocrine system being used to regulate homeostasis. Let's just verbally go over a couple things for a minute so I don't forget that. All right? Homeostasis is maintained through what process? Negative feedback. Right? Negative feedback. Does positive feedback maintain homeostasis?
No.
Absolutely not. Good. Negative feedback contains a set point. A stimulus is going to pull you away from the set point. That is going to be detected by some type of sensor. And then the response is going to bring you back to the set point, right? Negative feedback loops that maintain homeostasis are going to use the endocrine system and the release of hormones. We talked very specifically about regulating blood sugar. Blood sugar levels are maintained by what two hormones? Insulin and glucagon. Insulin and glucagon, right? You might even say that insulin and glucagon are opposing hormones, right? Because they are doing the opposite of each other. One is decreasing blood sugar, one is increasing blood sugar, right? Which one of those two hormones is responsible for decreasing blood sugar? Insulin. Right? But they are opposing or antagonistic hormones. That's it. You guys have just received 95% of the questions on the test. All right, let's stop right there. Are we good?